Here's a little exercise for you to practice working with the central dogma in molecular biology. So let's quickly remind ourselves what the central dogma is. So we start with the information for an organism stored in the DNA. That is the blueprint of life that contains all the information. This information in the DNA is then transcribed into a messenger RNA, and this messenger RNA is transported out of the cell and translated via ribosomes into proteins. So the flow of information goes from DNA to RNA to protein. And we know that the DNA is made up of 4 bases, A, T, G, and C, adenine, thymine, guanine, cytosine. The messenger RNA is made up also of 4 bases, adenine, uracil, which replaces the thymine, guanine, and cytosine. Whereas proteins are made up of 20 naturally occurring acids. And it's the sequence of the proteins and the amino acids in the proteins, rather, that determines the function of the protein. And the sequence of the amino acids is encoded in the DNA. So in this exercise, what we want to do is we want to do more or less break with the dogma. We want to do exactly the opposite. We want to take a protein sequence and figure out if we can find the corresponding DNA code for that. So here is an example. Write down your first name. So here the first name would be Angela. Now obviously this is just single letters, but as you know, we can also write amino acids in a single letter code. So here is the single letter code for amino acids. I've also included the 3-letter code. This is the letters in italics. So here, for example, this A in a protein sequence stands for the amino acid alanine, which is abbreviated by ALA. C stands for the amino acid cysteine, which in the 3-letter code is CYS, cis. For example, H is histidine, and which is abbreviated in the 3-letter code with HIS. And you get the gist of that. So what we are going to do now is we try to match our letters in the name with the amino acids. And we see that we've got an A at the beginning, so we know already that this stands for alanine. The second letter is an N, and we find this here as N is the abbreviation for asparagine. We then have a G. Here we have glycine. E stands for glutamic acid. L stands for leucine. And A, we have yet another alanine. So this is a very short protein, but we can use that very nicely to back or retranslate this amino acid sequence into a DNA sequence. How are we going to do that? Well, we can use our genetic code and our code table to do exactly this. So we have an A in the first place here, that is alanine. Now we need to find in our code table where we have this amino acid A. And we see we've got this here, ala, we've got the A's down here. And now we need to figure out what is the genetic code in the DNA that would give us this alanine here. Now we can just simply go to the left and we find in the table that the first position is a G. So we can write this down as a G. Okay, what is the second position? Because we know that we have triplets. The second position is we are just simply going up. That is the second position, and we find this is a C. And for the third position, we go to the right. And we find that this could be actually a T, C, A, or G. So we can write this as T, C, A, G. And while this is a little bit strange, we know that the genetic code is degenerated and that more than one codon can actually code for the same amino acid. And that is exactly what we see here. So the alanine is encoded by either GCT, GCC, GCA, GCG. So that was our first letter, that was the alanine. Now we can do exactly the same for the next amino acid, for the N, which was the asparagine. So we have to find the N in our table, asparagine, and we find that here, pretty much almost in the middle of the table. It is ASN is the 3-letter code. We've got N here. And now let's see what codon we use. So in the first place, we go to the left. We see it is an A. The second position, we go up and we find an A again. So adenine and adenine. And the third position, we go to the right and we find a T or a C. T or a C. So the amino acid asparagine, N, is encoded by AA, and then it can be a T or a C. Please be aware that we have sometimes the same letters for the amino acid and the bases. This is sometimes a little bit confusing, especially when we have A. It could be alanine or is alanine in the protein, but adenine in the DNA. If we've got G, this would be glycine in the protein or guanine in the DNA. C, in the protein this is cysteine, in the DNA it is cytosine. And we can also have a T, which stands for threonine, as the amino acid, or thymine in the DNA. So be careful, be aware of what system we are working in. Okay, now we've got our N. Let's do the next letter. So here we've got a G, and in the protein this is glycine. Where do we have glycine? We've got glycine down here, right in the bottom right-hand corner. So the first base would be a G, which stands for guanine. The second position, we go up, is also a G. again a guanine. And the third position can be again T, C, A, or G, highlighting the degenerate nature of the genetic code. So we've got half of it already. Now let's look at the next one. This one is an E, and E stands for glutamic acid down here next to the glycine. So glutamic acid, E. And again, we do exactly what we have done before. This would be a G in the first position. We go up. Now we've got an A in the second position, and we've got an A or a G in the third position. The next letter, we've got an L here, which stands for the amino acid leucine. And we've got leucine here again. Now this time we've got quite a few options to consider. So this could be, in the first place, it could be a T. Let's do the T first. We go to the left. So this could be a T. It could be in the second position, it would be also a T. And then we have the third position where we have an A or G. A or G. Or it could also be a C in the first position, because we see that the L is also encoded by a C. If we go up, we see the T in the second position. And then we've got TCAG for the third position. And finally, we do our A again. And we've done that already, so we can easily just simply copy that and put that into our sequence. So now we've got our nice sequence here, which corresponds to the DNA sequence. Now, of course, we would probably also indicate that the protein then stops after this sequence, and we would put in a stop codon. And this stop codon, we can check where we find a stop codon. So we've got 3 stop codons possible. So this could be TAA or TAG or TGA. So we could have TAA, TGA, or TAG. So this is our stop codon. And therefore, what we can do is we can now write basically the name that we have, and we could just, wherever we have this degenerated code, we just pick some that we quite like. So it could be GCC, AAT, GGT, GAA, TTA, GCC. Stop. So here you can write your name in a GNA code if we assume that this is the protein. I hope this makes sense, and thank you very much for watching.